Good afternoon and welcome back to the Mistra SafeChem final webinar of phase 1, or welcome if you are a new listener who have just logged on. This afternoon we have two sessions left on the agenda. The first one is about life cycle based assessment tools, and the second is about the Mistra SafeChem toolbox, an example from one of our case studies, and the next steps, a few words about the next steps towards a safe and sustainable chemistry. As usual, I want to remind you that we have a Q&A session at the end of this session, so please feel free to send your questions, enter your questions in the chat, and we will try to answer them as promptly as possible. With that, I would like to introduce the session number 3, life cycle based assessment tools, and the first speaker, Hanna Holmquist from IVL.
Thank you, John. So my name is Hanna Holmquist. I'm at IVL and I've been strongly involved in the work package focusing on research for life cycle based assessment tools for chemical assessment. I would like to give you an introduction to this session on toxicity impact categories in LCA or in life cycle assessment, and our research towards operationalizing tools for safe and sustainable by design assessment. First, I'd like to give you some background on life cycle assessment or LCA. So in LCA, we start from a function, and that could be provided by a product or a service. And for this function, we look at its full life cycle from cradle to grave. And for this life cycle, we quantify flows of raw material outtake and emissions. So we go from raw material outtake via manufacturing, packaging, use, and then towards end-of-life options. That could be recycling or disposal. And then we quantify these flows of emissions or raw material outtake. Another piece of background information relevant here is on the concept of safe and sustainable by design. In 2022, the EU Commission made a recommendation in December to establish a European assessment framework for safe and sustainable by design chemicals and materials. This was, of course, when the Mistra SafeChem program has already been running for quite some time, but we still see that our tools that we developed, they are fit for operationalizing this concept and to be applied in the assessment as described in the framework. So the framework comprises of 5 steps. 4 which are shown here: the assessment of hazard properties of the chemical or material, the human health and safety of production processes, the environmental and human health risks in the use phase, and then finally environmental impacts along the entire life cycle. So this image has a lot of information, and I will not talk through the whole thing because you will hear about many more details when my colleagues present in just a little bit. I just want to highlight that we have focused our research on enabling LCA that includes both human toxicity and ecotoxicity impact categories, and we have had a strong focus on the design stage. And in this research, we have involved researchers at IVL, at RISE, DTU, and of course our industry partners. So a huge thank you goes out to all the researchers who have been involved. Next up is the more detailed information about the tools that we have further advanced in our work. So my colleagues will give you talks on the new developments in USEtox and in ProScale. You will have a brief description of the tools and then focus on the advancements done in the program. And I encourage you to place questions in the Q&A so we can answer them at the end of the session. So with that, I welcome my colleagues Kerstin from Borries and Tomas Rydberg to give presentations. First up is Kerstin.
Yes. Hello, everyone. My name is Kerstin from Borries. I'm a PhD candidate at the Technical University of Denmark, and I'm here today in place of Peter Funke to talk about the new developments that we've made in USEtox, where Mistra SafeChem was one of the major projects that made these advancements possible. So to start off, what is USEtox? USEtox is the scientific consensus tool for characterizing human toxicity and ecotoxicity impacts of chemicals. It allows multi-pathway exposure assessment of both chemical emissions and now also product applications. And it comes with a large database for now almost 10,000 chemicals, which was expanded from previously around 2,000 or 3,000 chemicals. So how USEtox assesses chemical impacts is that we provide input data in either terms of chemical mass in products or chemical mass emitted to the environment. And then it does different fate and exposure pathways and assesses these, how they distribute in the ecosystems and the humans. And then finally, we arrive at risk and impacts measures for ecosystem quality and human health. So with the new advancements on the use stage exposure, we can now estimate consumer exposure, assessing and replacing chemicals in different types of product types. And it is built on the scientific foundation for comparative assessment of chemicals. So it allows both the traditional characterization factor derivation that we use for life cycle assessment, but also integrates a risk assessment perspective where we can calculate cumulative exposure risks and, for example, derive maximum acceptable chemical concentrations in a product. The new version of USEtox comes with several new consumer product modules, and we've integrated them in individual interfaces. So the original version and also the current scope of state-of-the-art LCA software is focusing on the direct emissions. Here we have just added a new sediment compartment for considering these emission pathways. And then we've added 5 different types of products, which is personal care products, building materials, children toys, indoor paints, and food contact materials. So to take a little bit deeper look at how these interfaces work, I've brought an example for you. Looking at hydroquinone in a shampoo. Hydroquinone is a topical skin bleaching agent and is typically found in shampoos at a concentration of about 1.3%. And it has been implicated both in terms of being irritating to the skin in humans, and also there has been some evidence of increased tumor incidence in mice. So it's definitely something where we would want to assess the overall impacts. So to do that, we actually only need to provide 2 major types of inputs. First, you need to select the chemical that you want to assess from the database and provide the chemical mass fraction in the product. And you need to define your product and usage characteristics for the product that you want to assess. And with just this information, USEtox then determines different types of results. It provides you with the exposure of the user and the general population. And then you get different metrics for the relative measure of risks, so something like hazard quotients and incremental lifetime cancer risks, and also the output we usually use for the LCA world is the characterization factors as a cumulative measure of impact. And you can also export some of the intermediary data, such as product intake fractions. So how do we provide this, and how does it look in the interface? You can see up here, we provide the CAS number and the chemical mass fraction, which is the 1.3% that I mentioned earlier. And then you select your product type, and many of the products have already predefined, including the product mass that is typically applied and the exposure duration. But you can also easily add your own product if you have some specific conditions. And then based on these inputs, USEtox calculates all the different results, including the exposure, the risk assessment, and the characterization factor. But before I dive into that, let's take a look at what is going on in the background of the model. So basically what we provided was the product usage and the chemical content. And with this, the USEtox basically calculates the inventory flow, let's say, so the chemical mass in the product. And based on the product category, it also knows the near field point of entry. So which compartment is this chemical emitted to? In the case of a shampoo, it's the skin surface area. And from there, the chemical distributes into different other compartments. Most of it will be washed out with the wastewater, of course, but some of it might be directly taken up into the skin of the user. And another fraction might also just evaporate into the near person air. And from these compartments, again, USEtox further models where the chemical goes in the far field environmental compartments, and how then at the end there will be a total intake towards the direct user, but also the global population. So how can you link this now with your standard LCA impact models? Well, in the LCA, usually in your inventory analysis, you provide emissions to air, water, and soil, and you come up with your inventory, which is your kilogram emitted per functional unit. Then you combine that with your characterization factor for each of these environmental compartments given in DALI per kilogram emitted, and you then arrive at a total damage in DALI per functional unit. Now for the product or use stage, we actually consider the whole product basically as an emission. So we have the emission given as a kilogram chemical in product per functional unit. And the characterization factor now also has a slightly different unit, which is DALI per kilogram in product. But if you combine these two, you actually end up with the total damage again in DALI per functional unit, which allows you to then accumulate or sum up all the different impacts across all the life stages and get an integrated result. Yes. So this brings me to my conclusions. You've seen USEtox 3.0 combines far-field and near-field exposure and accounts for multiple exposure pathway. You can use USEtox both for a risk and a damage-oriented approach, so to derive cumulative exposure risks, for example, also to make recommendations in terms of maximum acceptable chemical concentrations in products. But you can also use it to really integrate use stage impacts with the impacts from other life cycle stages in LCA. And the product interfaces allow you to easily input new products. And what I've not shown here today, but which is actually quite intuitive, you can also perform batch mode calculations to assess several chemicals at the same time. So with that, I would like to finish up here. If you have any further questions, please write them in the chat. And I will hand over the word to Tomas Rydberg.
Thank you, Kerstin. I will need my glasses to read my slides here. I'm Tomas Rydberg. I'm a senior advisor at IVL. I've been working with life cycle assessment and impact assessment for many years now, and I have been involved in this Mistra SafeChem program, particularly on the development of a method called ProScale, and I will get to that in very short. You see the rough agenda here of my speech, and I will talk a little bit about what we have done specifically in Mistra SafeChem. What is the idea behind ProScale? Well, it's similar to USEtox that Kerstin presented, in the sense that it presents a method that you can use to quantify somehow the impacts regarding human toxicity and ecotoxicity on products and value chains. We wanted to develop ProScale, let's say, from a very different starting point compared to USEtox, because we wanted to see what can be done with already existing ways of dealing with chemicals within the REACH regulation, for example, both in terms of how chemicals are conceived in terms of the hazard classification, but also in terms of the data that you can find in various contexts within the context of REACH. Therefore, I would like to spend a minute or two to discuss a little bit where we were when we started the project, basically, or Mistra SafeChem started. So you can see here a very schematic picture of a life cycle from left to right, and on the upper part you see in red the various arrows and pointing to worker exposure or user exposure, for that matter, or service life exposure. In Kerstin's vocabulary, that would be the near field exposure, basically. And when we started here, we had the scope of ProScale 1.5, which was the version of the method at the time, ProScale for human health impacts, that addressed specifically this part of the life cycle and the type of impacts relating to that. Whereas basically where USEtox 2.1 was when we started Mistra SafeChem was essentially to look into the environmental exposure, the ecotoxicity impacts, and the indirect impacts on humans, so the environmental mediated human exposure. And then what has happened partly during or in the time frame during Mistra SafeChem is that USEtox 3 has been developed, as we just heard, which also looks into this near field exposure situation, and also ProScale has come up with an additional module called ProScale E that will cover the environmental, or does cover the environmental exposure and effect, and could be used then for further extension into the indirect human exposure part, like this. So what are the constituents in our ProScale? Well, as I said, we start with, let's say, what is already there, but we also add a few things to make it compatible with LCA. The mass flow is essentially the life cycle as we know it, with the various flows in the system relating to the function of a product that is ultimately what we want to do with the value chain. And then we have some default settings in the method, and these can be essentially replaced if you have more detailed or more specific information. But for the mass flow, it's typically the bill of material, what we call it here. We have an exposure concentration factor, and of course this is not surprising that we need to include the exposure. So what we did here is that we looked at various existing tools for that purpose, and we found this USEtox. So that's the European Center for Toxicity, which is essentially an association with many of the big chemical industries as members, and they have a tiered risk assessment approach, TRA, and we wanted to build on that because it's also accepted in the exposure scenarios that are mandatory to report under the REACH registration regulation. Now, in order to match the mass flow together with the exposure concentration, we needed to also include a factor called the person-hour factor, or what we call person-hour factor, which is essentially a metric of the amount of working hours that are needed for each produced unit of product in a system, and at the instance where you are exposed to a certain chemical. In order to have a sort of a default or a reference number there, we lean on something called the best available technology reference documents, or BREF for short. So because there you can find how many people are working in a specific sector. So that's a good, let's say, generically available estimate that can be used in this kind of default setting. But again, if you are a producer of a chemical or you have an intended process for a certain production of a chemical, you can instead use that specific number if you want to. And then ultimately we have derived a numerical score based on the hazard phrases in the hazard classification of chemicals, and we combine that with let's say a modifier based on the occupational exposure limit, or sometimes the derived no effect limit, to arrive at a specific metric specific for each chemical, but here also specific to the specific process that the chemical is used in, as you can see. Another part of what we have actually done is that we've then tried out this approach on an indoor paint situation, and interestingly we started with a document where you could actually find something like a generic formulation of an indoor paint. So that would be from the product environmental footprint pilot phase, where they did a pilot scale testing on indoor wall paint. And I won't go into the details here, but basically this is then the mass flow or mass balance of the system that we studied, and we confined our analysis to the production of the chemicals and mixture of them into the paint, and included also the application, but not the use phase and the waste management, because we had to stop there to get it done in time. And we also, of course, included the value chains from the raw materials. And here you can see all the upper boxes that are essentially the raw materials, and these are the expanded value chains for these materials. So it's quite still a lot of process that we had to dig into actually to get this result in place. And the results per se are essentially there to try if the method works or not. So it could not necessarily be used to define if the indoor wall paint is good or bad or not. But you can see that the application stage is where most of the toxicity impact potential, or if you want to call it risk, occurs. And it is relatively, let's say, intuitive in a way, because that's when you can imagine, if you have painted a house or a wall or whatever, that you feel the smell of a paint, basically. So that's the signal. Now, what we also did is that we used all the information we had gathered in this paint study and added up with some other processes that we had assessed, and then we tried to look into the global scale of manufacturing in such processes and added all of that up to what we call a normalization score. So that's a kind of an estimate of the global impact potential as expressed in ProScale units, because that's a sort of information that LCA practitioners typically want to use in their assessments, and especially if you are looking into a certain type of aspect and comparing with another one, you may find it useful to have an external reference, and in this case then the global, or an estimate of the global impact in your specific impact category to compare with for the different impact categories. It turns out that there are basically 3 process types that are extremely important in this particular calculation. It's the crude oil refining and extraction, and it's steam cracking, which is more or less a very core process in the petrochemical industry. But of course, then we predominantly assessed processes in the petrochemical industry, so it might not be too surprising. But anyway, it's a value for normalization that you can actually use, both for inhalative impacts and dermal impacts. Now what we also did in Mistra SafeChem was to look into the need for this ProScale E, which is then the ecotoxicological extension of ProScale, if you might say so. And you recognize the structure of this picture, but it outlines instead the factors that we have come up with for ProScale E. So we have not the same factors, but we have similar functions. We have the mass flow, obviously, as before. We have now an environmental release factor. We have a degradation factor, and we have a hazard factor. So you recognize again the hazard factor concept, but here it's of course based on the H phrases for ecotoxicity. And it turns out that the hazard factor, both here and for the ProScale for human toxicity, can be very well predicted using the methods that you heard about in the morning. So that's very convenient, obviously. And here we have defined a set of default settings that you can use for this kind of assessment. And then we also, in parallel with the work that you heard reported this morning, we were looking at what would be the process specifically to explore or to fill the data gaps that we are in need of to do the ProScale assessment. And this slide is very difficult to read, obviously, but the thing is that you can do it in a kind of a stepwise approach. So you can assign the hazard phrase going from top to bottom. So if you have already found at, let's say, ProScale hazard class E that the substances you are assessing belong to that class, you can stop there because that is what will determine the score that we put on the substance, essentially. And then you can just follow it stepwise downwards. And having said that, I'd like to leave the word back to Kerstin again, who will give a more detailed presentation of some of her detailed work. Kerstin, it's all yours.
Yes, hello again. For those of you who haven't seen my earlier appearance, my name is Kerstin from Borries. I'm from the Technical University of Denmark, and I'm doing my PhD there. And today, in this presentation, I get the pleasure of taking you a little bit into the details of what I did during my PhD project. So to start off, we are facing a big challenge when we want to assess different chemicals, because there are around 350,000 chemicals registered for production and use on the market. But if we take the earlier version of USEtox as an example, we have around 3,000 chemicals that are fully characterized. Now with the newer database, maybe we go up to 10,000, but we see there is still a huge gap between what we would like to assess and what we currently can assess. And there are, of course, many different reasons for that. We do have quite a few chemical parameters that we require as input into our models, and this is true for both ProScale and USEtox. And typically, if you don't have all the information, you don't actually get a result. Then there is, of course, a limited overall data availability. This is related to time and cost-intensive testing and data confidentiality issues. And finally, there are several challenges in applying prediction methods, because often the applicability varies across chemicals and models, which makes it challenging to understand when which model is actually applicable for which chemical. And finally, we, especially in comparative assessment, we see some issues with integrating measured and predicted values due to their varying uncertainty. And we haven't really figured out yet how to do that so that the comparison is actually still fair. So the first question I wanted to address during my PhD project was, how can machine learning systematically fill chemical assessment data gaps for key input parameters across globally marketed chemicals? And to address this question, I started with 2 objectives. First, I wanted to prioritize key input parameters for machine learning. And then secondly, I wanted to assess machine learning potential to fill data gaps for those prioritized parameters across the global market of chemicals. So to do that, I started by selecting 38 input parameters. These were based on USEtox. And then I assessed each of these parameters based on two criteria. One, we had the characterization uncertainty. So that means how robust or how relevant is a parameter to obtain robust characterization results. And then the second criteria was the data availability, which describes the general suitability of applying machine learning. And combining these two criteria, I made then a prioritization for developing machine learning methods. And for those parameters that were prioritized, I then went further on to analyze the chemical space that is covered by the available training data. I jumped over one slide, sorry. So first we will look at the prioritization of the input parameters. You can see on the left the overall list of all the parameters we assessed, but we are zooming in a little bit on the two important groups. First of all, 13 parameters that were actually prioritized for machine learning. And you can see here in this column, you can see the visual representation of the characterization uncertainty. So how relevant is the parameter for obtaining robust results? And then secondly, the data availability. And obviously a larger symbol means more relevant or more data available. And when we were cutting off basically a certain threshold, then we prioritize parameters. But we also saw for some parameters where we had this high relevance for obtaining robust results, but the data availability was really, really low. So for 9 parameters, we flagged them with highly critical data gaps, which need to be urgently addressed, but maybe not with machine learning as the primary tool. So then we are focusing now on these 13 parameters that we prioritized for machine learning and wanted to look at this data availability. How representative is it actually for our overall space of marketed chemicals? And for that, we defined a target space. So here we are looking at 130,000 marketed chemicals. And this cloud you can read basically as a map. So every dot is one chemical. And chemicals that are very similar to each other are clustered together. So this you can also see that then these clusters pretty much align with common chemical classes. So you have some examples over here. For example, you have some steroids, and over here you have a cluster of amines. And principally, we can see that in this lower left area, we see all the cyclic and aromatic structures, while up here we have basically everything else. So any more chain-like molecules and anything that has maybe inorganic parts or metal organometallics. So this is the overall diversity that we need to represent, that our machine learning models need to understand in order to make robust predictions across this whole diverse space of chemicals. And so we went then on to see how well does the available measured data represent this diversity. And here we're looking at an example for non-cancer human toxicity effects via oral exposure. And you can see again this overall cloud. All the light gray dots are the same chemicals that you saw in our reference space in the previous slide. And then in black, you can see all those chemicals that actually have measured data available. So you can see again, they are kind of spread out across the entire space, but they are also maybe more clustered in some areas. And in order to get a bit more of a quantitative measure, we also defined an applicability domain, which covers all the chemicals that are pretty similar to the ones with measured data. And this is the ones that you see in purple. So these points are the ones that, or these chemicals are the ones that we would expect to be able to predict with a machine learning that is built on the available measured data. And so you can see it's a quite big improvement. We had 5,000 chemicals with measured data, and now we can predict almost 50,000 chemicals, or we would expect to be able to predict almost 50,000 chemicals. However, we can also see that's maybe 30% of the overall space that we are looking at here. So there is still more than 60% missing. And what we also saw is that when we look at the representation across the different chemical classes, there are some chemical classes that are quite well represented, like the lipids and hydrocarbons here, but others where the representation is really, really bad. So here we have at the bottom alkaloids and homogeneous metal compounds. So this means that especially for these chemical classes, we will really struggle to obtain reliable predictions. And this leads me to my next question. if we now have this overall chemical space, how accurately or how reliably can actually machine learning models predict this global market? And to look at that, we are taking one step back to first look at the challenges for assessing the reliability of machine learning predictions. The common approach is first of all to look at a few different metrics. Usually you have the mean cross-validated prediction error. And this is of course very useful. However, it is a mean, so it doesn't tell you anything about the uncertainty for across different chemical classes if it's varying. And also you only get this validated on a certain set of chemicals. So it doesn't tell you anything about the reliability of the prediction for chemicals that are very different from the set that it has been validated on. Secondly, you have the applicability domain. This is somewhat commenting some of the issues that we have with the mean cross-validated prediction error, where it tells you something about how similar is the chemical that I'm interested in to the training chemicals. But then again, it doesn't give you any information on the actual prediction accuracy, either for the chemicals inside nor outside the domain. And again, for this one, we have the confidence score, which is something like the prediction accuracy on the nearest neighbors. But there we also have the challenge that we don't actually know if the nearest neighbors are actually representative of the chemical we're looking at. So this makes it very challenging if you have these three different metrics to actually really interpret what is the reliability of the machine learning model prediction. And instead we propose that we want to do a full uncertainty quantification that includes the inherent data availability that comes from the data, something like uncertainty or variability across measurement methods, test conditions, interspecies variability. And this is the one that we don't really have any control over when we start building the model. And then the other type of uncertainty is the model-related one, which is something about how suitable is the algorithm that I'm using, how uncertain are the modeling parameters that I fitted, and did I choose appropriate predictor variables. So this is something where I might have some leverage on. And finally, we want this uncertainty quantification to be individual to every point prediction. That means the models have to be able to assign different levels of uncertainty to different types of chemicals. So this is something we addressed in my second project, which is to build uncertainty-aware machine learning to predict non-cancer human toxicity. And here I had also, again, two objectives. First of all, I wanted to identify and develop machine learning methods that allow this full uncertainty quantification. And secondly, I then wanted to apply this method to this broader space of 130 marketed chemicals to see how reliable is it and can I identify any chemical classes of concern. So we were looking at two major paradigms of uncertainty quantification in the machine learning world, which is conformal prediction and Bayesian neural networks. We combine two approaches here for their calibration for quantifying this full uncertainty, both aleatoric and epistemic uncertainty, and then use the better calibrated model to make this prediction of the whole chemical space with our 130,000 chemicals. So I will not bore you with the details of the calibration, but jump right into the results. If you're interested in the calibration and the more theoretical part, I encourage you to keep an eye out for my upcoming publication. But here again, we will look at this overall nice cloud of 130,000 chemicals. And now the color tells you something about, did the model predict a high or low uncertainty, where high uncertainty is the more orange colored dots and the lower uncertainty is the turquoise ones. And you can see that there are some different highlighted classes here. So we have a group of steroids that is quite toxically predicted. We have something that you might be more familiar with, polychlorinated types of cycloalkenes and also polybrominated and polychlorinated substances like dioxins. They are these number 2 and 5 and 6. And we also have organothiophosphate, which are often used as pesticides. And finally, we have a large cluster over here, which is different types of, or a large group of PFAS. And we can look again also at the uncertainty of the underlying prediction. And so again, you see this cloud with the color indicating how uncertain was the prediction that was made. And we can see that some of these high chemical classes of concern are also predicted with quite high uncertainty, in particular the steroids up here, and also the polybrominated and polychlorinated substances. And we can also see that the model is particularly uncertain in the mixed metal non-metal compounds and different types of alkaloids, which is interesting because this is what we also saw in our previous work, which is the ones that were least well represented by the available data. So this brings me to my conclusions. We prioritized 13 out of 38 parameters for developing machine learning, and we saw that there is a potential to predict 8 to 46% of marketed chemicals. However, we also flagged 9 critical parameters that have really critical data gaps and that need to be assessed urgently through other means. We demonstrated how uncertainty-aware machine learning can quantify prediction reliability. And this is, of course, not only interesting in order to understand how good is this prediction that I'm looking at, but it also allows us to propagate this uncertainty through chemical assessments. And this could facilitate a better integration of predicted and measured data. And finally, with the global chemical space predictions, we can identify chemical classes of concern and areas of high uncertainty, which allows us to target these areas to really make sure that we get most reliable predictions or improve the reliability of some predictions. So how do we do that? Looking ahead, we can see that there will be strategic efforts needed to systematically extend the capacity for reliable predictions, because we can see that we can predict maybe up to 50% right now, but the other 50% we will need to go more creative. And some of the ways that we see this might be done is through systematic data curation for underrepresented chemicals. We can do some transfer learning from related parameters or novel data types, and we might also be able to do the hybrid modeling to integrate mechanistic knowledge. So these are some of the things I hope that we might be able to address in our second phase of Mistra SafeChem. But for now, I will hand over the word to Anna-Karin and Hanna Holmquist.
Okay, my name is Anna-Karin Hellström, and I'm from RISE Research Institute of Sweden, and I will present this session together with...
My name is Hanna Holmquist, and I'm from IVL, and I've been co-leading this work package on life cycle-based assessment tools together with Anna-Karin.
And the title of this session is How can the life cycle-based assessment tools be used? Mistra SafeChem case studies illustrate operationalization. So here we will describe a little bit how USEtox and ProScale has been applied in our case studies. So in 2021, we foreseen that USEtox and ProScale could be applied in 3 different contexts. In the chemical alternatives assessment, which focus on substituting hazardous chemicals in a specific product or process applications, the importance of including life cycle considerations in this methodology. And Peter Fanke and co-workers published a framework in 2020 with the title of Life Cycle-Based Chemical Alternatives, Life Cycle-Based Alternative Assessment for Chemical Substitution. And this framework is developed to be aligned or be able to use within the chemical alternatives assessment. It can be used in the tools can be used in chemical footprint assessment, where we focus on assessing toxicity and ecotoxicity profiles of product or processes. And here we are referring to human toxicity and ecotoxicity impact categories in LCA. The chemical footprint assessment can also be, of course, included in a full life cycle assessment, where we also integrate other impact categories. So here is we present the case studies that we have been working on. These case studies most of them have already been presented this morning. And in the figure to the left, we have mapped the different case studies along the life cycle, starting from the raw material extraction, the production, the use phase, and end of life. And in case study A, we focus on process optimization for extracting cellulose nanocrystals from post-consumer cotton. This is still at the lab bench. It's at a low technical readiness level. In B, which was presented by Berlin this morning, they wanted to replace palladium as a metal in the hydrogenation reaction with nickel and electrochemical process. In C, safe and sustainable by design in discovery chemistry. Here in silico predictions has been used to derive characterization factors with USEtox, and here more than 100 of substances has been assessed, and this will be more described by Hanna in a second. Then we have, we leave the lab bench in D, production of low-risk building block chemicals for surfactants. Here, the focus was on process optimization or upscaling processes. And then we have 2 substitution case studies, one focusing on the indoor air quality in a car cabin, and the other one focusing on substitution of cyclic siloxanes and silicones in cosmetic. And this one will be presented in more details by Lisa Skedung in the next session. So if we shortly just deep dive into a few of the case studies. This one was presented by Maria in the last session before lunch. And what they wanted to do was to extract cellulose nanocrystals from post-consumer cotton by using citric acid hydrolysis. So what you do is that you have your cotton garment, cut them into pieces, and they're added into a high concentrated solution, water solution of citric acid. The citric acid could be recovered by evaporation and recrystallization. You also get some, of course, waste here. And then the nanocellulose could be filtered off. And here we used the cradle-to-gate analysis since the potential application was not yet defined when this study was performed. It was used to support decisions on the process design at the low technical readiness level. And the data that was used was generated from the laboratory tests. And the framework developed by Piccino was used here, which is kind of a semi-quantitative way approach of using process calculations. And here we just show that the citric acid had the highest contribution to 14 out of the 16 impact categories.
So with that, we have another illustration from our case study work, and that is a case study that we call the Safer and More Sustainable by Design in Discovery Chemistry. For those of you who listened into session 2 this morning, Per-Olof Syrén presented this case study. Herein, we have made selection of so-called SafeChems by filtering on intrinsic hazard, but we've also been assessing the impact potential of these chemicals based on the USEtox derived characterization factors. Both of these workflows have been high throughput and based on in silico based approaches. So we've been able to cover the many chemicals, as Anna-Karin just mentioned. To the right, you see an illustration of the workflow coming from the selection of the enzyme through filtering substrate and products and then arriving at this list of so-called SafeChems. In this graph to the bottom here, you see the characterization factors as a result. To the left, it's very small, but to the left is the ecosystem characterization factors, the ecotoxicity ones, and to the right is the human toxicity ones. And you see on the x-axis is the factors for emissions to fresh water, and on the y-axis is the emission to fresh water, and on the x-axis is the emission to air. And on the ecotoxicity side, they align fairly nicely, and we have a larger spread on the human toxicity side, probably illustrating the more intricate fate processes involved in the exposure of humans. So here, by using the USEtox derived characterization factors and assessing the potential impacts, in addition to these intrinsic hazards, we could identify additional chemicals that had a fate who increased the associated potential impact with these chemicals, for example, if they accumulate in the environment. And also, of course, we will link to this case study a LCA where we compare the biocatalytic route with the more conventional process route. And then, of course, we can apply the characterization factors in that LCA to include the toxicity impact categories. Another example is the study that was presented by Belén Martín Matute, the sustainability assessment of a novel hydrogenation reaction process. Here you see some preliminary results. We also covered the 16 impact categories from the environmental footprint method, as Anna-Karin just showed. And herein we have 5 of them extracted in an illustration. In this study, we have a nickel catalyzed electrochemical reduction of alkynes to Z-alkenes and alkenes to alkanes in a water-based reaction medium. We have scarce metals that are replaced by more abundant metals and materials, and we have hydrogen gas from fossil resources replaced by water. And The LCA, which was a screening type LCA, cradle to grave, was used to find further optimization possibilities. And we also in the case study was involved a non-target screening approach and hazard assessment in combination with this LCA. And you can see here in the illustration that, of course, we had a look at the resource use, as that was one of the drivers for the case study. And as has been mentioned already, the LCA made it possible to identify the platinum electrode as a potential hotspot. It can be reused very many times, but replacing it could be beneficial for the further sustainability of this process. The LCA, and then with that, the LCA results will, of course, improve for the nickel-based system, but we can also see that the LCA makes it possible to avoid trade-offs. So we can look into several other impact categories than those that were driving this case study. But the most important result is, of course, that the LCA spur research on alternative electrode materials.
So some key takeaway from this first phase of Mistra SafeChem. Risk assessments and life cycle assessments are complementary, but there is room for further alignments. And this was briefly showcased in the biocatalysis case study. Process design can tackle resource use and related emissions, while the intrinsic hazard of substances or mixtures can only be eliminated through chemical substitution. And there is a need to further explore how to integrate USEtox and ProScale in the safe and sustainable by design. And that will be a focus on phase 2. We also see that there is a need for education and training for the use of these tools and how to interpret the results. And adopting an integrated effort through transdisciplinary collaboration is essential. So in summary, using an LCA approach in the early design stage can help quantify the impact of the design goal and identify environmental hotspots for potential improvement. Here, we have had a strong focus on the toxicity and ecotoxicity indicators, but of course, a broader scope assessment is important to also include other impact categories so you don't shift the burden to another impact category. And then finally, the inclusion of life cycle thinking in the chemical alternatives assessment we see also is crucial.